Okay, so our next speaker is Timothy Yu, a graduate student from Dr. Jesse Bloom's lab at the Hutchinson Cancer Center, presenting about measuring viral escape from polyclonal antibodies. So please welcome Timothy.
Awesome. Thank you for the kind introduction. Thank you for this opportunity. I'm really excited to tell you about some of our recent work on this idea of being able to measure viral escape from polyclonal antibodies. So I want to start by talking about one of the main reasons for why it's really hard to develop an effective flu vaccine, and it's that flu rapidly evolves to decrease antibody neutralization. So what I mean by that is conceptually, flu has this viral protein called hemagglutinin, and it uses this protein to be able to get inside and infect cells. And so when we get vaccinated, our body will produce polyclonal antibodies that can target the HA protein at multiple distinct epitopes. And so any of these antibodies can neutralize the virus and prevent infection. Now the problem is, of course, that over time, flu gains mutations in HA. And so at some point, we no longer have these antibodies that can recognize these epitopes. And so this is a phenomenon known as antigenic drift. So now in our lab, we specialize in using deep mutational scanning to study antigenic drift. And so far, we've actually measured the effects of all single amino acid mutations to HA on antibody neutralization. The issue is that these studies don't really tell us the full story. And that's because you can imagine with polyclonal antibodies, if you have a single mutation, you might knock off antibodies binding one epitope, but you're still going to expect to have antibodies that recognize the other epitopes and can neutralize the virus. And so in that case, we're not really fully capturing the conditions of antigenic drift. So because of that, we need to extend these DMS studies to multiple HA mutations. And so one of the other primary reasons we need to do this experimentally is that because everyone knows that mutation effects can combine in very complex ways, and so you can't simply just add up the effects of single mutants that we have from our prior studies to try and predict what happens in this multiple mutation case. And so our overall goal was to be able to do this combinatorial deep mutational scanning and use models to try and predict which combinations of mutations will escape antibody recognition ahead of time. And hopefully that will be useful in being able to design better flu vaccines. Okay, so I'm going to talk about this deep mutational scanning platform that we have now. So this was developed by a graduate student in our lab named Francis. And so the way this platform works is we start with a library of mutagenized influenza HAs. And so these are HAs that have multiple mutations, each associated with a unique barcode. And we can use PacBio long-read sequencing to match the variants to the respective barcode. We can then take these mutagenized HAs and assemble them into a library of influenza virus variants. And so one thing we also do at this stage is add a distant H6 virus. And so the reason we spike this in is because H6 viruses are really exotic, and so nobody in the human population should have antibodies against this H6 virus. And so this is something that we can use as a neutralization standard for comparison later. So with this library of viruses, we first incubate our library with human sera, and so these contain the polyclonal antibodies that can target the virus and neutralize it. And we also do a mock selection control. So this is just the viruses on their own without the sera. We then try and infect cells with these populations, and then lastly, we sequence the barcodes of the viruses that are successfully able to replicate in these cells. And we do this with the Illumina short-read sequencing. And so in the very end, we end up with something like this. So for each variant in our library, we have the counts, the barcode counts in the sera population and the barcode counts in the mock selection control. And we can normalize each of these counts to that H6 neutralization standard. And so then we take these normalized counts in sera and normalized counts in mock, and we can get this ratio that we're going to call the escape fraction. And so the way to interpret the escape fraction is that if it's 1, it means that the variant was completely able to escape the antibodies in sera. And if it's 0, that means that the variant was completely neutralized, so it was not able to get into the cells at all. Okay, so now that we have these escape fractions, we have to think about how do we actually analyze this data. And so in DMS, we have a bunch of sequences, we know what their mutations are, and we have this measured escape fraction, right, at some sera concentrations. And so there are many ways we could try and model this relationship between sequence and function, and we decided to go with an approach that we thought would be the most interpretable, which was this biophysical model. And so what we wanted to be able to do was learn what are the specific epitopes being targeted by neutralizing antibodies in the sera, and then secondly, what are the mutations that prevent these antibodies from being able to recognize the epitopes. And so just to go into the details of this model now, so we start out with a few assumptions. The first assumption is that each antibody can bind only one epitope. So like a blue antibody can only bind the blue epitope. It can't bind blue and red, for instance. And also antibodies bind independently without competition here. And so this is just to make it simple, so like blue antibodies can't compete with red antibodies to bind their respective epitopes. So with these assumptions, we can define the first part of our model. So on this left-hand side, I have the escape fraction that is measured in DMS at some sera concentration. And on the right-hand side, we're going to model this escape fraction as a product over this variable I'm going to call U sub e. So this is going to represent the fraction of time that the epitope is not bound by antibodies at some sera concentration. So I think this is best illustrated with a simple example. So imagine that you have mutations in the blue and orange epitopes, and they completely prevent antibodies from binding. So in that case, the fraction of time that the epitope is not bound is going to be 1, because antibodies are always unbound. Let's say that in the unmutated epitopes, the green, red, and purple epitopes, antibodies are bound, let's say, 50% of the time. And so the probability that the fraction of time that those epitopes are not bound is going to be 0.5. And so then the escape fraction that we would observe in DMS is just going to be a product over all of these unbound fractions. And so in the end, we would, let's say, in this case, it would be 0.125. So the next thing we want to do is take this U sub e term and try and translate it to a more biophysical level. And so to do that, we had to make a few more assumptions. The first were that all antibodies targeting a given epitope have the same affinity. So all of these blue antibodies, for instance, that target the blue epitope, they have the same affinity. And also there's no cooperativity in antibody binding. And the reason this is important is because we wanted to take this fraction of time that the epitope is not bound by antibodies and translate it over to something more biophysical. And so we do this through a Hill function. So we model as a Hill curve. And so we can relate this U sub e variable to this new variable that I'm going to define as phi sub e, which is going to represent the total binding activity of antibodies to an epitope. So you might be wondering, so what does this phi sub e term actually represent? So this total binding activity of antibodies to an epitope, it's composed of 2 different terms. And so these are terms you can get by rearranging the Hill equation in the previous slide. So the first term is going to be the affinity of antibodies to the epitope. And then the second term is going to be the abundance of antibodies targeting the epitope. So I think a nice intuitive way to understand how this works is, let's say you want to increase the binding activity of antibodies to the epitope, you would want to make this phi term more negative, right? So then there are 2 ways you can make it more negative. You could either increase the affinity of antibodies to the epitope, so you make this delta G term more negative, or you can increase the abundance of antibodies to the epitope. So in that case, you just increase this F term right here. And so in practice, we're not going to be able to resolve these specific contributions on affinity and abundance given our phenotype that we're measuring is neutralization. But what we can do is do something like in Julia's talk where you assume that mutations have additive effects on the free energy of binding. And so we can express this total binding activity right here as a sum over the effects of specific mutations on antibody binding at each epitope. And then there's this other bias term that we think is really important. And these are going to represent the activity of antibodies binding an unmutated epitope. And so this is because you can imagine that, let's say we don't have mutations at all, antibodies are not necessarily going to be bound 100% of the time to these epitopes, right? They could be bound 20% of the time, 50% of the time, 80% of the time. So this is that term that's going to control for that. Okay, so just to summarize this model, right, we start out with these observed escape fractions in DMS. We model them as a product over these antibody unbound fractions. And then we can take these unbound fractions at each epitope and relate them to the total binding activity of antibodies via this Hill function. And then we can take this total binding activity of antibodies to each epitope and express that as a sum over the effects of individual mutations. And so basically in the end, we want to go from this escape fraction in DMS to learning these beta parameters down here, which represent the effects of mutations at specific epitopes, right? And so again, how do we sort of visualize these beta terms, right? So these beta terms, again, right, they represent the effects of mutations on antibody binding at each epitope. And so let's say we had a toy example right here. And here I'm showing, like, let's say in the top, you can see in this blue heat map that any mutation at site 4 is affecting escape of antibodies towards the blue epitope. And so in this case, that means that if you have a mutation at site 4, antibodies are no longer able to bind the blue epitope. And the same logic for the red epitope beneath as well, except for a different set of mutations. And so one way we can summarize this heat map is by plotting the total site-wise escape. And so here what I'm just showing you is just a sum total of all the effects at a specific site. And so, for instance, you could basically just sum over each of these columns, and that's what you're getting in this plot on the right. And so the reason I show this plot on the right is because you're going to see a couple of examples of these in the next few slides. Okay, so now we can actually talk about actual experiments. And so the first experiment we did was with making a combinatorial DMS library in the background of a recent H3 flu virus. And so this was the Hong Kong 2019 variant. And so we had 3 to 4 mutations on average in our library in this Hong Kong 2019 background. And so the next thing we needed to do these experiments was actually find a human sera sample that actually neutralizes this Hong Kong 2019 variant. And that's really important because what we're looking for is what mutations or combinations of mutations then decrease this neutralization that we see from the sera sample. And so with these 2 components, we can then do this experiment. We got the escape fractions, we fit the biophysical model, and we were really excited to see what happens. And it ended up looking like this. So this was slightly disappointing to see. So it's just a single epitope, and it's escaped by a single site at site 189. And so the reason I want to highlight this is because this is something that we do expect to see occasionally. And that's because we know that site 189 is within the receptor binding site of the HA. So we know it's heavily targeted by antibodies. Another thing we know is that some individuals can produce these highly immunodominant antibody responses against single epitopes. And so I think this really highlights why flu is such a big public health problem, because this person went from having a super potent neutralizing antibody response against this flu, this Hong Kong 2019 virus, and then if that Hong Kong 2019 virus just gains a single mutation, that person loses that neutralizing antibody response and becomes susceptible to infection. And so just to convince you that this model is actually working, just look at the raw escape fraction scores, and you can see that basically any variant with this mutation at site 189 is going to escape. Okay, so of course I told you we were trying to model multiple mutations and also the effects of mutations at multiple epitopes, right? And of course with our previous example, that's not the case, right? We actually can't tell if it's working. And so fortunately we had another library in our lab, and so this one was the same type of library but done in the pandemic H1N1 HA background. And so this was a Michigan 2015 strain. And so the special thing about this strain is we had a set of monoclonal antibodies that we knew targeted 2 distinct epitopes. We had prior structural studies that showed that. And so in doing so, we realized that we could use these monoclonal antibodies as sort of a positive control for our model, because by definition, a monoclonal antibody targets a single epitope. And so what we did was we did the selections, and so we just wanted to verify that if we fit our biophysical model on this, we get a single epitope first of all, and then also we get the exact escape mutations that we would expect from this monoclonal antibody. So this is exactly the case for this first antibody. So this was 2D01. It's escaped by mutations at site 130, 156. And then we had another monoclonal 6649. This one targets a different epitope and is escaped by mutations at site 164. And this was just to see that, with the monoclonal antibody, right, we should only find one epitope, and that's exactly what we found. So the next thing we did was do the exact same experiment, but this time with pooled human sera. And so the reason we did pooled human sera is because this is expected to be polyclonal, because you can imagine we're taking antibodies from multiple people and combining them together. And so what we were trying to see is, can our model actually figure out that there's multiple epitopes being targeted? And that's exactly what happened. We found that there are 4 epitopes being targeted by the antibodies in the sera, and they're escaped by these particular mutations. And something is very interesting if you look a little closely at these escape profiles, is that if you look at epitope 1, this escape profile actually looks a lot like that 2D01 escape profile for the monoclonal antibody. And if you look at epitope 4, it also looks very similar to 6649. And so we thought this was very nice to see, and we think these results suggest that the epitopes that are learned by our model are actually real. And so we can take this complex problem of taking someone's sera and asking, what are the types of neutralizing antibodies that this person has? And so we can use this deep mutational scanning approach to actually figure that out and be able to define, what are the specific antibodies in this person's sera that target specific epitopes. So I want to conclude with a little bit of a summary and also some future directions. So this is very preliminary work, but I hope I've convinced you that we can model viral escape using combinatorial VMS data and interpret that at the epitope level. Another thing that we can use this model for is actually for prediction. And so when a new viral variant appears, we can actually use this biophysical model to predict the likelihood that it's going to escape antibodies in an individual's sera. So you can imagine if some individual has antibodies that target 4 different epitopes, right, and we see this new viral variant appear, and it has a mutation in one of the epitopes, right, that person is probably going to be fine, and that's what our model would predict. But you can imagine if there's some viral variant that appears that has mutations at all of the epitopes that this person targets, right, then that person might be in trouble. And so some of the next steps, so we're actively trying to scale and profile a lot more sera samples. And so, I've showed you some very limited data in the previous slides, and one of the things that we think is going to be very important is this person-to-person variation in epitope targeting. And so the more sera samples that we test, the better idea we can get of how this, people's antibodies repertoires and this epitope targeting looks like at the population level. And then, of course, then the goal would be to then apply the results of these models to try and improve flu surveillance and vaccine selection. And so one thing I wanted to mention just to conclude is that this approach is general, and we can apply it to other viral DMS systems as well. And so, for instance, like HIV envelope and SARS-CoV-2 spike. And so there are people in our lab that are working on these projects as well. So with that, I will conclude with some acknowledgments. Thank everyone in my lab, and also some of our awesome collaborators at Fred Hutch. So yeah, and I'm happy to take any questions.
Hi, great talk. This looks really exciting. I was really struck by I think it was the H1N1, where you find like 4 different epitopes. The sequence, the maps onto the in general, the maps onto the sequence look pretty sparse of the effects on across the sequence, which is kind of what you kind of hoping to see, right? But I guess I was wondering, from the modeling standpoint, it seems like you're fitting a lot of parameters. Are you enforcing that sparsity through the model fitting procedure, or I guess more generally, can you explain a bit more about how you're doing the quantitative modeling? Do you have to worry about overfitting, and do you have to manually enforce sparsity?
Yeah, so it's a great question. So we do actually manually enforce sparsity, so we have a lot of regularization in place, and so that just prevents there from being too many redundant beta effects. One key thing about how we're fitting these models, right? So we enforce this regularization, but also what we do is we just iteratively fit models until basically you have a redundant epitope appear. And what we define as a redundant epitope is that there's just basically all 0 beta effects. And also one of the key things is that that activity term that I briefly mentioned, that is actually super positive, and actually the interpretation would be that there's basically, if there were antibodies targeting this epitope, they're never bound, right? And so that's the key sign that we know we have hit the limit for the number of epitopes that's being targeted in the sera sample, and of course on top of that we have that regularization as well.
Okay, thanks.
Same question. Thanks.
Okay.
This is a clarification question. So if mutation is present, let's say in a certain location, let's say in epitope A, you assume that escape is due to antibody binding to epitope A, correct?
Yes, that's right.
So how do you deal with LS3 then?
Sorry?
Or LS3?
Yeah, so we would expect to see allosteric, and this is why, so one other key point about how we fit these models, we were thinking about enforcing some sort of spatial regularization, so basically any site that's far away is basically not going to be allowed to be in the same epitope, but we don't enforce a spatial regularization just because we expect there to be, for instance, allosteric effects, it might occur, and so that would appear in our model. So you could have 2 mutations that are in sites that are far away from another, but if they are affecting the same epitope, they would appear in the same epitope.
So you do have monoclonal single antibody assays for each mutation, data for that is already available?
Well, we can do these experiments, so same combinatorial DMS library, but with a monoclonal antibody.
So you have those, and you incorporate the data into your model?
We're not incorporating that. So when we fit the model for the sera, we're fitting just to the sera alone.
Okay.
Yeah, and we have a question from the online audience. So the question is, what's the length of barcodes in mutant library design, and are these random barcodes?
Yeah, so 16 nucleotide random barcodes.
Awesome, thanks.